Every single second of every day, trillions of microscopic chemical reactions inside your body generate hazardous metabolic waste products. These reactive oxygen species and unstable free radicals constantly threaten to damage your delicate cellular membranes and mutate your DNA. If left unneutralized, this persistent oxidative stress damages mitochondrial machinery, degrades structural proteins, and accelerates physical aging across all tissues. Yet under healthy biological conditions, human cells maintain a remarkable degree of resilience against this relentless microscopic bombardment. This crucial cellular defense is coordinated by a single master antioxidant molecule known to modern biochemistry as glutathione. Unlike ordinary dietary antioxidants like vitamin C or vitamin E, which you must consume from food, your body produces glutathione directly inside every living cell. It is concentrated in exceptionally high amounts inside your liver, which operates as the primary biochemical filtration facility for your entire bloodstream. Whenever environmental pollutants, prescription medications, alcohol metabolites, or industrial chemicals enter your circulation, glutathione neutralizes them. In the liver, detoxification occurs in two distinct phases to ensure harmful foreign compounds are safely processed without damaging surrounding tissue. During Phase 1, cytochrome P450 enzymes chemically modify fat-soluble toxins, often temporarily creating intermediate compounds that are even more reactive. During Phase 2, glutathione steps in to bind directly to these reactive intermediates through a specialized enzymatic process called conjugation. This vital conjugation reaction transforms dangerous fat-soluble toxins into stable, water-soluble metabolites that can be safely excreted through your kidneys and bile. Without adequate intracellular concentrations of glutathione, this second phase stalls, allowing reactive metabolic intermediates to accumulate and damage liver cells. The unique molecular architecture of glutathione is composed of three specific amino acids bonded together in a precise sequence: glutamine, glycine, and cysteine. Because it consists of three linked amino acids, biochemists classify glutathione as a tripeptide molecule. The functional centerpiece and primary reactive engine of this tripeptide is a specialized sulfhydryl chemical group attached to the cysteine amino acid. This active sulfur hydrogen bond acts like a biological electron donor, eagerly transferring electrons to unstable free radicals before they can attack cellular structures. When glutathione neutralizes a free radical, it donates an electron and temporarily becomes oxidized into a compound known as glutathione disulfide. In a healthy, metabolically active cell, an enzyme called glutathione reductase immediately captures this oxidized molecule and recycles it back into active form. This continuous enzymatic recycling loop requires cellular energy in the form of NADPH, directly linking your metabolic health to your antioxidant defense capacity. When your cells produce ample energy through healthy metabolism, over 90% of your intracellular glutathione remains in its active, reduced state. However, when chronic oxidative stress exceeds cellular energy production, the active pool of reduced glutathione is rapidly depleted. Scientific research demonstrates that as humans age past the age of 30, the body's natural production of glutathione begins a steady, progressive decline. This biological decline is significantly accelerated by modern lifestyle stressors, including chronic sleep deprivation, refined dietary sugars, psychological stress, and alcohol. When glutathione concentrations fall below critical physiological thresholds, the mitochondria inside your cells begin leaking excessive reactive oxygen species. This state of mitochondrial dysfunction triggers premature cellular senescence, where aging cells cease dividing and secrete inflammatory signals into surrounding tissues. Recognizing the indispensable role of this master molecule, many health-conscious individuals attempt to replenish their levels using standard oral glutathione capsules. Unfortunately, conventional oral glutathione supplements suffer from notoriously poor bioavailability when swallowed by humans. The strong hydrochloric acid in your stomach and powerful digestive peptidases in your small intestine rapidly dismantle the delicate tripeptide bond. By the time the digestive process is complete, virtually none of the intact glutathione molecule survives to enter the systemic bloodstream and reach target cells. To bypass this severe digestive barrier, medical researchers and nutritional biochemists have focused on providing targeted precursor compounds. The most thoroughly researched and clinically proven precursor molecule is n-acetylcysteine, commonly referred to worldwide as NAC. Cysteine is the universal rate-limiting amino acid required by human cells to synthesize new glutathione internally. While your body generally maintains an abundant supply of glutamine and glycine from dietary protein, free cysteine is relatively scarce and unstable. N-acetylcysteine overcomes this transport challenge by attaching a stable acetyl group to the delicate cysteine molecule. This protective chemical modification shields NAC from digestive degradation, allowing it to be absorbed efficiently into the bloodstream through the intestinal lining. Once inside your cells, cellular enzymes cleave away the acetyl group, liberating pure, bioavailable cysteine for immediate glutathione synthesis. Clinical human trials confirm that oral NAC supplementation reliably and significantly elevates intracellular glutathione concentrations in liver, lung, and brain tissues. In hospital emergency medicine, high-dose intravenous NAC has served for decades as the standard frontline antidote for acute acetaminophen toxicity. When an overdose of pain medication exhausts hepatic glutathione reserves, intravenous NAC rapidly restores liver antioxidant pools, preventing irreversible liver failure. This life-saving clinical application provides undeniable medical proof of how effectively NAC acts as a direct biochemical substrate for glutathione production. Beyond acute hospital care, maintaining optimal glutathione reserves provides comprehensive protection across every major physiological system in your body. Because the human brain consumes 20% of the body's resting oxygen while possessing modest antioxidant defenses, it is uniquely vulnerable to oxidative damage. Glutathione actively shields delicate neural synapses from lipid peroxidation, supporting cognitive clarity, executive function, and long-term neurological resilience. In your cardiovascular system, glutathione prevents the dangerous oxidation of low-density lipoprotein particles inside delicate arterial walls. By neutralizing free radicals within the vascular endothelium, it preserves healthy nitric oxide production, promoting flexible blood vessels and optimal blood pressure. Your immune system is equally dependent on abundant glutathione reserves to mount an effective defense against environmental pathogens. White blood cells, including T lymphocytes and natural killer cells, require adequate intracellular glutathione to multiply rapidly and coordinate targeted immune responses. In addition to targeted amino acid supplementation, specific dietary choices provide the essential elemental building blocks your cells need to synthesize glutathione naturally. Cruciferous vegetables such as broccoli, Brussels sprouts, kale, and cauliflower contain rich concentrations of beneficial organosulfur compounds. Broccoli sprouts in particular provide dense amounts of glucoraphanin, which your body converts into the bioactive phytochemical sulforaphane. Sulforaphane acts as a potent natural activator of the NRF2 genetic pathway located inside the nucleus of your cells. When the NRF2 pathway is activated, it signals your cellular DNA to upregulate the expression of the enzymes responsible for synthesizing and recycling glutathione. Furthermore, the biochemical enzymes that recycle glutathione require specific trace minerals to function with optimal catalytic efficiency. The critical recycling enzyme glutathione peroxidase is fundamentally dependent on an adequate dietary supply of the essential trace mineral selenium. Incorporating selenium-rich foods, such as Brazil nuts, wild-caught fish, and pasture-raised eggs ensures that this antioxidant recycling loop operates smoothly. Another valuable synergistic nutrient is alpha-lipoic acid, a unique antioxidant capable of functioning in both water-soluble and fat-soluble biological environments. Alpha-lipoic acid helps regenerate depleted intracellular glutathione by directly recycling vitamin C and vitamin E reserves throughout your tissues. Dietary glycine, found abundantly in bone broth, gelatin, and collagen peptides, ensures that your liver never runs short of the second structural amino acid. Daily lifestyle habits also exert a profound influence on preserving your existing antioxidant pool from unnecessary depletion. Regular moderate aerobic exercise and structured strength training stimulate an adaptive hormetic response that permanently enhances your basal antioxidant enzyme expression. Prioritizing consistent, deep restorative sleep allows your brain's glymphatic system to clear accumulated metabolic debris while cellular antioxidant reserves replenish. Conversely, excessive alcohol intake places a severe metabolic burden on your liver, forcing it to burn vast quantities of glutathione to clear toxic acetaldehyde. Minimizing exposure to environmental toxins, such as tobacco smoke, heavy metals, and industrial chemicals helps conserve your body's natural defense systems. By combining a nutrient-dense whole food diet, targeted precursor support with NAC, adequate trace minerals, and restorative lifestyle practices, you can effectively optimize your cellular defense. Maintaining robust intracellular glutathione levels provides your body with the biochemical armor required to maintain energy, protect organ function, and thrive long-term. As medical science continues to reveal the central importance of cellular defense in healthy aging, supporting your master antioxidant remains an essential pillar of health. Please remember that this presentation is strictly for educational and informational purposes and does not constitute individualized medical advice. Always consult with a qualified healthcare professional before starting any new dietary supplement regimen.